Thanks very much, Ian. Yeah, I'd also like to start by thanking Ian and Frank and everybody else for the opportunity to talk to you today. So I'm working in the HipTox program, ably run by Gordon here, where our role is to try and understand how environmental pollution affects the pathogenesis of disease with a focus on the brain. And what I want to talk a bit about today is our experimental approach. We have a great team here today in the audience as well from Scotland. And so I want to tell you about the model systems we are using to try and understand how environmental pollution causes disease at a mechanistic level. And some of it is quite biologically complex, but I hope you can bear with me. So we'll see if I go forward or backwards. So my research career has been, in total, trying to understand how environmental agents cause human disease and the metabolic processes that have evolved to protect our cells from the deleterious effects of environmental chemicals. These are not just important in defining how the pathogenesis of disease and the environment in which we live, but they're also critical in defining how we handle therapeutic drugs, because the same enzyme systems and pathways that define toxicity and efficacy for environmental agents do the same for drug molecules. So, as I just said, these enzyme systems I'm going to describe to you today are of critical importance defining the survival and death of cells due to environmental factors. They're probably also key in defining susceptibility to diseases such as Alzheimer's diseases, as Paul has already described. By the way, I'm a bit worried about the car parking allergy because I've had that problem for many, many years. Anyway, so next slide. So what we've been doing, we carry out in vivo studies because the physiological processes that define toxicity are very complex, and although you can get really great information from iPS cells or in vitro test systems, understanding the physiology of what the relationship between exposure, innate immunity, toxicity is in essence too complicated to just work out from in vitro studies alone. So we've been developing ways of monitoring how cells respond to environmental stress in an in vivo context using mouse models. So the first part of my talk, I won't get to the second part. Ian's already told me off for having too many slides. Francisco was unfortunately, but fortunately for him, has just taken up a senior lectureship back in Spain. He's played a key role in this work. Colin Henderson, who was my partner in developing all the model systems we've developed over the years, is now retired, unfortunately. Mike Mann has left. Nobody's around anymore, apart from Phoebe, who's tolerated me for a bit at least. So anyway, so that's our collaborators. I put this up because of work that I'm not going to be showing to you, but I won't get to. But this is a request that we have a big collaboration on the application of humanized mice, a mouse humanized for pathways of drug disposition with Tres Cantos, which is the unmet clinical need development of drugs for malaria and Chagas disease and things. And they asked me to put the slide up, which is quite appropriate, I have to say. So the premise of our work, which we've been going on now for many years, that adaptation of cells to our environment is key in cell survival. You get exposed to something that is toxic, you activate detoxification pathways, you activate DNA repair pathways, you activate inflammatory response pathways. And in principle, these allow you to survive a hostile chemical or inflammatory environment. However, when these are overcome by too much exposure, or indeed perhaps by chronic exposure over long periods, these pathways no longer protect cells, and when they get overridden, cells die, and you end up with a toxicological response. So over the years, we have made a lot of reporter mice, and I'll explain what that is in a minute, that say when you're exposed to something, which pathway alters in which cell type for how long and what exposure level. This is useful for risk assessment relating exposure to disease risk, but also tells us about mechanisms. So when an oxidative stress pathway is activated, and that's very early on, followed by a DNA damage response pathway later on that tells us the key initiating event is oxidative stress, followed by DNA damage. If you look at a patient, it's all happened. So you don't know cause or effect in that context. So I won't go into these because there won't be no dinner tonight, so I'll carry on and show you some examples of how we do this. And this is a nice slide from Fran. So in our hip tox program, we have really been focusing on 3 or 4 different pathways. One of them is regulated by a receptor called the AH receptor. This is a pivotal receptor in defining human and animal responses to polycyclic aromatic hydrocarbons, aromatic amines, a whole plethora of toxic and carcinogenic environmental chemicals. This system is also now being shown that work that Gita Stockinger did and others at the Crick is also a key regulator of innate immunity. So activation or interaction with this receptor, you can interpret it as an early event and a potentially deleterious event. NRF2, which is a key regulating orchestrator of cellular responses to electrophiles and oxidative stress, as you heard, banded around ad libitum with virtually every mechanism of toxicity. And the other one is the DNA damage response modulated by the gene P53. So let me just give you some examples how these reporter systems work and how you can use them to understand toxic mechanism. I'll try and go through this quickly. So the AH receptor binds a polycyclic aromatic hydrocarbon, gets activated, goes to the nucleus, and then activates genes like cytochrome P451A1, which in principle results in detoxification and removal of these inert chemicals from the body. However, this enzyme can also catalyze the metabolic activation of polycyclic aromatic hydrocarbons to toxic electrophiles, which are carcinogenic and overtly toxic. So what happens when you expose a reporter mouse to a polycyclic aromatic hydrocarbon? Now, all we've done to generate a reporter mouse is that you take the gene, and it's regulated through these elements in the promoter of the gene, but instead of substituting the CYP1A1 gene with a reporter which generates a color or something else you can measure easily in vivo, either in tissue sections or non-invasively, and I'll show you some of that in a minute. So this is a mouse, a reporter, a CYP1A1 mouse, in work I did many years ago with John Clark and at the Roslin Institute. And this is an untreated mouse, a mouse not treated with a polycyclic aromatic hydrocarbon. And in this reporter mouse, when you give a polycyclic aromatic hydrocarbon, you get this massive increase in the activity of AH receptor signaling. And so this is the skin and the hair follicle. This is the liver and the centrilobular hepatocytes. And interestingly, even in the gallbladder epithelium, highly sensitive to aromatic nitro compounds, to carcinogenicity, like benzidine, that used to be found in the leather dyeing industry. This enzyme activates those aromatic amines to carcinogenic products. So you can see that in this reporter mouse, you get really detailed analysis of where your receptor is activated, when it's activated, and you can relate that then potentially to toxic mechanisms. It also provides you with biomarkers that says these environmental chemicals generate these responses. Another interesting example, acetaminophen, paracetamol, is not toxic until it is metabolized in the liver to an imidoquinone, a highly electrophilic toxic metabolite. This is when it's still the most common cause of suicide in the UK, paracetamol overdose. It's metabolized by the cytochrome, keep getting the wrong button, P450 system to this, which is detoxified by glutathione. And a long time ago, we knocked out this enzyme, glutathione transferase, which in theory should have increased the toxicity of paracetamol, because we predicted that this reactive intermediate would accumulate. And we got it completely wrong. When we knocked out this gene, these mice became completely resistant to paracetamol overdose, another potential treatment that we never really followed up on. So in the GST pie normal mouse, so these are animals treated with paracetamol. In the normal mice, you get this centrilobular liver necrosis in the liver, lipid accumulation. But in the GST pie null mouse, there's no effect at all. But when you then look at ALT, a measure of hepatotoxicity, in the wild-type mice, you get this huge increase indicating liver damage, and in the knockout, you don't see this anymore. So we crossed this mouse, the pie null mouse, into an oxidative stress reporter mouse. And when you do that, what is really interesting and tells you mechanistically what is going on is that here we have the wild-type mouse where you see the induction of oxidative stress using a heme oxygenase 1 reporter animal. In these central cells in the centrilobular hepatocytes are basically dead, necrotic, but there is, on the periphery, you're still getting live cells where you have an oxidative stress response. In the GST pie null mice, you're not getting any dead cells, but you're still getting the oxidative stress, which tells us that mechanism by which this enzyme is protecting against paracetamol toxicity is not because it's preventing oxidative stress, and we have evidence to show that it is also modulating a key signaling pathway associated with potential cytoprotection. So the other way you can use these reporters is to identify clinical intervention. So if you know a stress response is important in disease, you can get an early biomarker. So instead of having a model for motor neuron disease, which I'll still show you in a minute, and wait 40 weeks till you see whether your clinical intervention works, if you have a report that shows something already at 2 weeks, that has a massive effect, consequence for your science and for the identification of antidotes. And so here, the classical way you treat paracetamol overdoses with N-acetylcysteine, which scavenges the imidaquinone. And so this is just paracetamol-treated mice, and this is mice treated simultaneously or subsequently with N-acetylcysteine, which completely attenuates the toxic response to this molecule. And this treatment saves lives, of course. So we've made some more sophisticated reporter models where we've actually can measure a number of different endpoints in real time in the transgenic mice. So we can, this is a bit too complicated for today, but we can measure in situ. We can measure, as you've shown you, LAC-Z, blue staining when the stress is activated. We can also, we've made in the same model, we put in an excretable biomarker, which is human choriogonadotrophin. So when that is produced, it's excreted into the serum. So you've got a biomarker that allows you to track toxicity in real time. And we've also put in a fluorescent marker so that you can stick these mice under a camera, and without killing them at all, you can actually see when a stress response is activated and how that changes or is attenuated over time. So for any clinical or any intervention study, all you have to do is either measure a serum biomarker or put the animals in a scanner. So just to I'll go through very quickly of some of these reporter systems. I might actually skip these. We made these. So metallothionein 1 is a reporter that actually detects heavy metals. We're not using it in the HIP tox program, but it has obviously, if you're going to start talking about dust from brake linings and anything else, it might be an interesting model to use. Just showing plus or minus treatment with the metal cadmium chloride, this massive increase in the liver in reporter activity. There's a superoxide dismutase reporter as well. I think that's probably vascular activation. Totally different cells of activation of the superoxide dismutase reporter as from the metallothionein one. I'm going to skip some of this. This is just showing the real-time imaging of the calcium, of the cadmium chloride exposure, showing wild-type mice, and then you see this huge increase in luciferase signal in the livers of cadmium chloride-treated mice, indicating the activation, in this case, of oxidative stress in the liver of these animals. So a couple of things on the brain, as this program talk today is about the brain, in theory, not from me so much. So Douglas Vasey with Bruce Whitelaw, we created a DNA damaged P21 mouse many years ago. And we said, well, can you start measuring toxicological responses in the brain using this P21 mouse? And so what Douglas did, he treated etoposide, as do many anti-cancer drugs, alter cognizance in cancer patients. So anti-cancer drugs definitely have neurological effects. This is LPS, an inflammatory agent, and this is mercuric chloride. And what is interesting without going to other tissues, is you can see profound induction of this, in this case, DNA damage responses in different brain regions by application of these different things. So you can say, you can start dissecting out these tissues. We can look for other metabolic effects that might occur then. We have a particular interest with the Edinburgh team, Richard Meehan and Sari Pennings, on looking at epigenetic changes. These are non-mutational changes in DNA methylation that can generate really important biomarkers for epidemiology studies. Ian, you have to tell me when to shut up, by the way. I'm only halfway through the first half of this first half. This is not, I've got lots of anecdotes about over-speaking a meeting. So much where the whole audience fell asleep, actually. But so we crossed our P21 reporter mouse into a P53 null mouse and showed this whole of the activation of the P21, which was P53 dependent, because all the signals we saw were these things in the brain went away. This is just some other examples. One really interesting one is the superoxide dismutase 2 gene. Superoxide dismutase 1 was the first inherited susceptibility gene associated with motor neuron disease, highly topical at the moment. And when you actually get superoxide 2 activation in different regions of the cerebellum and other parts of the brain, you give these animals diazepam and that staining goes completely away. So it's actually removed the oxidative stress that has caused this constitutive activation. This is another study where we've put, where a German collaborator group put a contaminant, a food contaminant into the mice, and again showed very specific neurological changes in oxidative stress in the brain. So bear with me, Ian. A couple more, just to finish with a couple of studies where we've actually been using the reporter mice to understand neurodegenerative or degenerative diseases in general. So one is that we've been studying progeria, which is an early onset aging disease, as you will all know. It's due to the formation of a compound called progerin, which is a truncated version of laminin A. And that accumulates in cells and causes this, the onset of this disease. So we crossed a mouse model susceptible to progeria to our stress response models. And this is work done by Fran, which has been published. And progeria is associated with stress and degeneration of vascular smooth muscle cells. And what you can see using the hot reporter mouse is that when you have these mice that are homozygous for this progerian accumulation, you get this huge induction of oxidative stress in vascular smooth muscle cells, not only in the aorta, but in other cells in the heart. And these are mice that are homozygous for this, metabolic defect, and they have quite a severe phenotype early on. But even when you do this over time, using heterozygous mice that don't have this phenotype, we also see these increases in staining in smooth muscle cell, in vascular smooth muscle cells relative to controls. So here we've used these reporter mice to tell us about what happens in cells in relation to susceptibility to an inherited human disease. We see the same for the DNA damage response, with a massive increase again in the aorta at 30 weeks of age in mice that are heterozygous for the progerin gene. And I'm rushing through this a bit, but I wanted to get to the brain at the end of my talk. So the other study that we'll be doing has been looking at motor neuron disease, and we've crossed the reporter mice into models of motor neuron disease. And motor neuron disease, the actual mouse model, is generated by overexpressing this protein TDP43, which is associated with MND in people. And we crossed that mouse into an oxidative stress model and a DNA damage model. And what is really exciting in this study is that whereas nearly all the studies, the hypotheses around motor neuron disease are related to changes in the spinal cord, when you take our oxidative stress reporter and cross it into a mouse susceptible to MND, the major changes that we see, I think Phoebe carried out this work, are in the Purkinje cells and cells, what we believe are basket cells, and these are key regulators in the motor neuron system that could open up new avenues for trying to understand what causes the disease and ways of intervening. So I think I shall finish there. I think, Ian, was that all right? Oh, yeah, you're over there waiting to come. No, let me just read the conclusion. Stay where you are. So obviously, what I hope I've shown you today, I haven't shown you what the HipTalks Meet program is about. We're really excited about its engagement and the multiplicity with Paul and with the behavioral people and Gordon with his inhalation skills in coming up with something that tells us mechanistically how environmental agents affect the brain using these types of model systems. Our collaboration with Edinburgh is of key importance in our program because they are looking at epigenetic changes that environmental agents cause from taking the mouse tissues to study, which we believe will generate and hope will generate biomarkers for particular types of environmental exposure, so you can use those in epidemiology studies to classify what is there and what the exposures might be. So reported models are a powerful approach to define mechanism of toxicity. I hope I've shown you that. Because they preempt overt physiological and pathological changes, you can do studies in dramatically reduced time frames. They give mechanistic insights, as I've said, whether it's oxidative stress, DNA damage. The HO1 reporter also tells us about the function of the innate immune system. I can't tell you about that, but I think that in the end of the day, I think the application of these models hopefully will give us further insights in this complexity of environmentally induced diseases and point us in some directions to generate the sort of biomarkers that you need in the future. Thank you very much.